Hello guys, welcome back again in another video tutorial for microbiology with Shagnik. So welcome in another video of our cell biology lecture series. So today we will going to talk about a very important component of the cell, that is mitochondria. So let's get started with the video. So we have discussed or we have studied the mitochondria in our earlier times, and today we will going to just recapitulate them, right? And not only that, I am also going to provide some extra information about the mitochondria that you may probably not studied when in your earlier times, for example in class 11 or 12. So let's get started with the video. So first, a very basic introduction, that is the mitochondria that we are going to talk about are membrane bound organelles, and it is usually consist of two type of membrane. One is the outer membrane, another one is the inner membrane. Okay, and this membrane bound organelles is responsible for generation of chemical energy. Okay, this chemical energy that is generated by mitochondria is responsible for running all the important biochemical process in our body, and this chemical energy will be stored in a special chemical molecule called ATP, right? And that's why we are calling mitochondria as the powerhouse of the cell, right? So now we have to learn what are the important function of mitochondria. Is it is only for the generation of energy, but also it have some important tasks. So there are various important tasks that mitochondria play. For example, mitochondria play a very important role in cell signaling, in cell differentiation, in cell death. Okay, not only that, it play a also very important role in maintaining the cell cycle. Okay, and the biogenesis of mitochondria is somehow coordinated with those biological processes where mitochondria play a very important role. And that's why any kind of disorders in mitochondria will going to be very dangerous for that particular individual. There may be cardiac dysfunctions, mitochondrial diseases, as well as there are various important type of threat that is associated with any kind of problem in mitochondria. Okay, for example, heart failure, autism, mitochondrial disease, cardiac dysfunction. So it is very clear to us why mitochondria is very important, right? Now this mitochondria is consist of five important parts. One is the outer membrane that generally covers the mitochondria, and this outer membrane consists of various type of porine proteins. That means it allow passage of wide variety of molecules, right? And for larger proteins, it have various type of proteins. For example, VDAC, that is voltage dependent anion channel, that allow various type of nucleotides ions to enter in the inner membrane space. And inner membrane space is generally the space between the intermembranes. Sorry, it is not inner membrane space, it is intermembrane space. And this intermembrane space is generally the space between the outer membrane and inner membrane. And the layer or the membrane which is present in the inside is the inner membrane, right? Now, interestingly, this inner membrane has foldings, okay, or this inner membrane is compartmentalized into various type of foldings, and these foldings are called as cristae. Okay, now the question is, why this type of foldings? Now, at the very beginning of the video, I have told you that the main purpose of mitochondria is to generate chemical energy, which will be stored in a special type of chemical molecule called ATP, right? And if you know about the oxidative phosphorylation, especially the function of ATPases, which have dual function of ATP hydrolysis as well as ATP synthesis depending upon the situation. So this ATPase require a good amount of surface area for synthesis of ATP. Okay, if surface area is very high in amount, then there will be high amount of ATP production. So that is why this type of foldings are observed in the inner membrane, because remember, inner membrane have this enzyme ATPase. Okay, and these ATPs require a good amount of surface area for production of ATP. That's why those type of foldings are observed in inner membrane, and we will call this folding as cristae. Okay, not only that, this cristae also stuffed with some rounded like structures, and we will call them as F1 particles. We will going to talk about F0 F1 particles in details when I will going to upload the video of ATPase, a very important part of oxidative phosphorylation that I have not uploaded till now. We will going to learn about that F1 particle in that particular video. So for now, remember that this inner membrane have foldings, that is for increased surface area, and this increased surface area is important because inner membrane have ATPases, and this ATPases is responsible for ATP production, right? So that's the idea. Now, inner membrane covers the space which we will call as matrix. Okay. Now this matrix contains various type of things. For example, ribosome, tRNA, as well as DNA. Now the question is, in every eukaryote, DNA is specially and separately stored in nucleus, right? We all know that very careful, very wellly, right? So why mitochondria have DNA of its own? What is the reason behind that? And not only mitochondria, let me clear that chloroplast also contain DNA. So what is the reason behind this? So the reason is explained by a special and very important and interesting theory, that is endosymbiotic theory. According to this endosymbiotic theory, this mitochondria have prokaryotes as their ancestors. Okay, and those prokaryotes somehow become permanently fused with eukaryotes in distant past. Okay, and from that particular time, these prokaryotes remain as mitochondria in those eukaryotes, and that is the reason why today we can easily respire. Okay, and that is the endosymbiotic theory. So what endosymbiotic theory tells us? Endosymbiotic theory tells us that the mitochondria have ancestors who are prokaryotes, especially free living prokaryotes. These free living prokaryotes in the earlier times somehow becomes fused with eukaryotic organisms and remain as mitochondria inside it. And similar is applicable for the chloroplast also. And that is the reason why mitochondria have all the necessary things for its own survival. Okay, that's the idea. And that is why mitochondria have DNA. Okay. And this matrix also contain ribosomes because it have to synthesize proteins, right? So that is the idea. So now, interestingly, the protein and phospholipid ratio in outer membrane and inner membrane significantly differs. In outer membrane, the protein and phospholipid ratio is near about 1:1, and which is usually observed in any type of cell membrane. But inner membrane, you will get a higher amount of protein. Okay, and in matrix, 2/3 of the protein present in mitochondria will be in matrix. Okay, and this is very important to learn. And there are various type of biosynthetic processes that occur in specific region of the mitochondria. For example, in inner membrane, I have just told you there is ATPases, right? That is involved in ATP production. Not only that, the redox reaction of electron transport chain is also occurring in inner membrane because of the presence of various type of proteins involved in that particular processes. Okay. In outer membrane, there are various type of protein present that involved in various type of important processes. For example, the elongation of fatty acid. And for that particular process, there is a presence of enzyme called fatty acid CoA ligase. Not only that, there are oxidation of epinephrine is going on in the outer membrane. Okay. Not only that, there is also oxidation of pyruvate and other type of oxidations going on inside the matrix. So you can understand various type of processes is going on within the mitochondria, right? And what is the reason behind that? The reason is the proteins responsible for those particular processes are present in various type of regions of mitochondria, right? So we will going to see about them one by one. So first, outer membrane. So outer membrane is 60 to 75 angstrom thick, and it generally covers the mitochondria. And as I have just told you, it has a protein to phospholipid ratio similar to cellular membrane. Okay, and it contains a large number of porine proteins. I have also told you this, and I have also told you about the voltage dependent anion channel VDSE. This VDSE is responsible for entry of nucleotide ions and various metabolites from cytosol to inner membrane space. Okay, and remember, due to the presence of this VDSE and other porine proteins present in the outer membrane, the composition of small solute in the cytosol and the composition of the intermembrane space is same. Okay, only the larger protein require a specific N-terminus signal sequence for its entry. Okay, that is why the composition of larger proteins in cytosol and inner membrane space differs. Okay, and the larger proteins only get the chance to enter in the mitochondria if it has a signaling sequence at their N-terminus, and this signaling sequence will going to bind with the translocase, a large multiprotein complexes, and if that particular protein has this signaling sequence, that translocase will going to allow it to enter in the inner membrane space. Okay, and various type of mitochondrial proteins are also present that require specialized translocation complexes for their entry. Okay, so you can see various type of biochemical processes going on in outer membrane. Elongation of fatty acid, this is due to fatty acid CoA ligase, oxidation of the epinephrine, and degradation of the tryptophan because of the presence of monoamine oxidase, rotenone insensitive NADH cytochrome C reductase, kynurenine hydroxylase, and fatty acid CoA ligase. Okay, and if somehow disruption of the outer membrane occurs, then all the protein present in the inner membrane space will be liberated in the cytosol, causing the death of that particular organism or particular cell. Okay, so that is the outer membrane. Very simple. It have porine proteins and voltage dependent anion channel that allow entry of various type of solute in the inner membrane space, as well as the outing of various solutes from the inner membrane space into the cytosol. And remember, this voltage dependent anion channel remain as beta barrels that spans the outer membrane. But larger protein require specific N-terminus signal sequence for their entry, and they also require translocase protein present in the outer membrane for their entry in the inner membrane space, right? Various type of biosynthetic processes are going on, for example elongation of the fatty acid, oxidation of the epinephrine, and the degradation of the tryptophan, and monoamine oxidase, rotenone insensitive NADH coenzyme cytochrome C reductase, kynurenine hydroxylase, fatty acid CoA ligase are the enzymes that are the present in our outer membrane, right? Now let's move to the intermembrane space. This intermembrane space is generally the space between the outer membrane and the inner membrane. I have told you that this intermembrane space have same composition of smaller solute with the cytosol. And what is the reason behind it? The reason is presence of various type of porine proteins in the outer membrane, right? But the composition of the larger protein is significantly different between this intermembrane space and the cytosol. And what is the reason behind that? The reason is larger protein requires specific N-terminus sequence for their entry, right? Which is not present in all proteins present in the cytosol. An important example of larger protein present in the intermembrane space is cytochrome C. And we also call intermembrane space as perimitochondrial space. Now let's move to the inner mitochondrial membrane. Inner mitochondrial membrane have various type of proteins that responsible for electron transport chain redox reaction. ATP synthase is present, which is responsible for ATP production in the matrix, and specific transport protein that regulate metabolite passage into and out of the mitochondrial matrix. Okay, I have also told you that inner mitochondrial membrane have higher proportion of polypeptides, right? Now, a very interesting thing, that is inner membrane have specific type of unusual phospholipid, cardiolipin. Okay, and this cardiolipin have four fatty acid chains instead of 2. Okay, and this cardiolipin is responsible for impermeability of near about all type of molecules. Okay, and that is why all the proteins require specific membrane transporter for their entry into the matrix. Okay, so this cardiolipin or the presence of cardiolipin in the inner membrane generally act as a impermeable barrier. Okay, and its disruption causes various type of clinical disorder including neurological disorder as well as cancer. Okay, so due to the presence of this cardiolipin, remember inner mitochondrial membrane is very impermeable, and for entry into the matrix all protein require specific membrane transporters. Okay, so I can say that the concentration of the protein in the matrix will be significantly lower, right, as compared to inner membrane space, and their composition or their types will also be significantly different, right? So that is the inner membrane space, and this is the structure of cardiolipin. I have also mentioned 4 fatty acid present instead of 2. Now the cristae. Cristae is the compartmentalized area of inner mitochondrial membrane, which is especially a fold. Okay, so inner mitochondrial membrane have numerous compartmentalized folds that we will call as cristae, and this cristae is especially responsible for ATP production because this cristae provides a high surface area, right? That is the idea, and that is the reason why the amount of cristae in various organisms significantly varies. Okay, not only that, the amount of the cell the amount of cristae present in cells also significantly differ. For example, let me consider a cell which is present in a particular organ that do not need a high amount of ATP production. Then the particular cell will have mitochondria with lower amount of cristae. Similarly, let's consider muscle cell which requires a high amount of ATP production have a higher amount of cristae. That's the idea. Okay, not only that, similar cell or a single cell can have different number of mitochondria, because at the very beginning of the video I have told you that liver cell have 2000 mitochondria. So I can assume that a cell should have, or a cell may have more than one mitochondria, right? So it may also possible, suppose a cell require a particular mitochondria for high amount of ATP production, then that particular mitochondria will have a larger number of cristae than other mitochondria present in that single cell. Okay, so simply as saying that the number of cristae and the number of folds will be depending upon the requirement of ATP production. If that particular mitochondria require a larger amount of ATP production, then simply that cristae will be present in numerous number. No doubt about this. Okay, and these folds are also stuffed with small round bodies. We will call them as F1 particles. Okay, so that is the idea of cristae. Now, the most inner part of the matrix, or the most inner part of the mitochondria, is the matrix. Okay, inner membrane space or inner membrane covers the matrix, and this matrix consists of 2/3 of the total protein in the mitochondria. This matrix has various type of enzymes, ribosomes, tRNA, and several copies of mitochondrial DNA genome, which appear as a nucleoid. Okay, now we all know that eukaryotic cell have nucleus in which DNA is present, right? Whereas in bacteria there is no nuclear membrane spaces, right? And in bacteria the DNA will be appear as a nucleoid, right? So it also important evidence of our endosymbiotic theory, right? So that is the concept of our matrix. So if I want to sum up this video with a very short conclusion, then the mitochondria is responsible for chemical energy production in the form of ATP, as well as cell signaling, as well as cell death, as well as the regulation of the cell cycle and various important things. They have 5 important layers. One is the outer membrane, which have various type of porine proteins, voltage dependent anion channels, which are responsible for entry of various type of solutes. But this outer membrane have translocases, which only allows larger protein having special N-terminal signal sequence. Okay, so we can say outer membrane is highly permeable, and they have protein to phospholipid ratio 1:1, right? Now after outer membrane we have inner membrane space. Before that, outer membrane play a very important role in various biological processes. For example, fatty acid elongation, then oxidation of the epinephrine, degradation of the tryptophan, depending upon the presence of their respective enzymes. Now in inner membrane space is the space between the outer membrane and inner membrane. They have similar composition of solute with the cytosol, but they have different composition of larger proteins, right? And what is the reason behind this? The reason is the presence of N-terminal signal sequence. So an important example we have learned is cytochrome C, right? And then in the inner membrane, which is very important, is responsible for various type of biological processes, and they have higher amount of protein to phospholipid ratio, right? And this inner membrane have specialized folding, and we will call this specialized folding as cristae. This cristae is responsible for ATP production. It generally provides a high amount of surface area, right? And it is stuffed with round bodies. We will call it as F1 particles, and at the end matrix that consists of genomes as well as proteins, right? And in that particular part, oxidation of the fatty acid as well as citric acid cycle is going on. Okay, so that's the overall idea of our mitochondria. So I hope this video will be helpful for you. Thank you.